Hello everyone. Today, we'll learn about the characteristics of modern selective biocides. The selectivity of a biocide typically refers to its biological characteristics that cause varying activity across different organisms. Modern selective biocides are those developed since the 1970s with high selectivity and safety, including carboxylic anhydrides, organophosphates, benzimidazoles, pyrimidinidiol, decarboxymides, benzamides, thiazoles, ergosterol, biosynthesis inhibitors, carbamates, substituted urea, phenylpyroles, anilynopyrimidines, methoxyacrylates, and cyanoacrylates. Compared to traditional multi-action site biocides, modern selective ones offer many unique features and advantages. Fewer or single action sites with varying selectivity across different organisms. Traditional multi-site biocides target multiple sites, some of which are shared by pathogens and crops or other beneficial organisms, posing a risk of phytotoxicity or environmental harm. Modern selective biocides target fewer or single sites, which are found only in some pathogens, not in other pathogens, crops, or other beneficial organisms, leading to selectivity between pathogens or between pathogens and other organisms. Currently, most modern selective biocides used in agricultural production are selective in nature. The selectivity of these biocides is influenced by differences in receptors or target structures in different organisms, the level of biocide accumulation in different cells of organisms, the abilities of compound activation, the abilities of biocide deactivation, or the genetic regulation of receptors in cells. Most modern selective biocides have systemic or local movement properties with curative effects, while many biocides for external use cannot penetrate plants after pathogen invasion to exert biocidal effect. Most modern selective biocides can penetrate plants to inhibit or kill pathogens. This is what we call systemic activity. Based on the strength of systemic activity, biocides can be divided into 4 types. 1. Local systemic. Once absorbed by green plant tissues, the biocide moves over short distances via the transpiration stream or between cells. An example is simoxanil. 2. Semisystemic or translaminar movement. Biocides move within the leaves without relying on the transpiration stream. For example, methoxyacrylate biocides such as azoxystrobin and cresoxymethyl. 3. Upward translocation. Once absorbed by a plant's roots, biocides are translocated upward via the transpiration stream. Most systemic biocides exhibit this upward translocation. Examples include benzimidazoles, carbendazim, ergosterol biosynthesis inhibitors, triadimethyl, benzamides, and metalaxyl. 4. Genuine systemic. When applied to a plant's roots or leaves, biocides can be translocated both upward and downward. Biocides like this are rare. For example, aluminum triethyl phosphate. Some modern selective fungicides only work where they're applied or before pathogen invasion, providing only protection. So it should be noted that not all modern selective biocides have curative effects. For example, most decarboxymide biocides are non-systemic and provide only protection. But these biocides work especially well on gray mold, indicating their selectivity. That's it for today. You can refer to more sources to learn more about the characteristics of modern selective biocides. Thank you.